some of our work that we do at TraitGenetics for plant breeding in companies and for the general plant breeding community in various different crops. So I will talk today a little bit about optimized marker sets for genotyping, how we do that it is a continuous process, and what are some of the outcomes that we have. But before I do that, of course, I'll give you a little bit of overview about TraitGenetics as a company. We are a company who has a motto to develop molecular markers and marker-assisted technologies that are specifically tailored to the breeding companies that we collaborate with. And we also provide services to use such technologies for accelerating the breeding process. We have a number of efforts. We're mostly focused on plants, and that's on the one side using all the available technologies from whole genome sequencing to the targeted analysis of genes or genomic regions to identify polymorphisms in genomes of any size. We also develop, of course, following up these things, genotyping arrays that can be used for the breeding process. Also in the targeted crossing schemes, we provide marker analysis services for the typical plant breeding processes such as marker-assisted selection or marker-assisted backcrossing. We also work, and I'm not going to tell you anything about that, about the identification of markers linked to specific traits. And we also screen mutagenized populations for SNPs or mutations. And of course, to a limited extent, we also provide consulting. I want to focus specifically on arrays, and I just have put up the technologies we're using. We have already heard about those things. We use both the Illumina and the Affymetrix technology for genotyping. We have a long tradition to work with the academic community in marker development, so we have been involved in the development of the wheat genotyping arrays, cotton, brassica, maize, tomato, sunflower, barley, and pepper. Of course, we have also developed additional proprietary markers. We are also working with publicly available SNP genotyping arrays that have been developed by others. So just to get back to the arrays, initially there were a lot of arrays developed for specific crops containing large numbers of molecular markers, but when people started to use those arrays for the analysis of molecular markers, they have quickly realized that some of the markers are of dubious quality, some other markers create redundant information. And in that way, there was quickly a demand from the breeding community, especially, to have arrays that are as good as possible, as cheap as possible, and as reliable as possible. So the need in the optimized marker area is to have only high-quality markers on the respective array. Plants have an unpleasant feature, and that is that mostly of them are polyploids, and in that way you have different genomes to deal with, so the analysis is much more difficult than in animals who are nicely diploid species. You want to have markers that are informative in as much material as you can have. They should not create all the same information. They should create specific information that is useful and not that much redundant information. Of course, ideally, you also want to have markers on the arrays that are diagnostic for specific traits or for important genes that control specific traits. Of course, price is always an issue in the breeding sector. And the data should be in a way gathered that they can be compared between different laboratories, and the system should be scalable for these things. I also want to just have one example that I'm going to present, and that's wheat. So I'll go through the different processes. So initially I start with version 1 of the optimized wheat array we have developed. That was about 2.5 years ago. So the way you start is, on the one side, you need genotyping data from as many lines that represent the breeding pool or the natural or the international breeding pool of the material. So initially, the work started by genotyping more than 2,000 wheat lines from different sources. With the wheat 90K array, which was at that time available, and internally developed 12K array. And based on that, we had subsets of markers, 26,000 that are polymorphic and useful for European elite material, for North American and Australian elite material, and for worldwide material, which we have analyzed together in collaboration with CIMMYT. So out of the about 100,000 markers, 33,000 markers were are polymorphic with an allele frequency of more than 3%. The other thing you have is the marker quality, and you see 4 different patterns. The typical pattern that you have in animals and in diploid species is this. You have very well separated clusters, but in the polyploid species like wheat, where you have 3 different genomes, the A, B, and D genome, you very often detect not only one genome, but you detect 2 or 3 genomes, and when you do that with the Illumina technology, you get more condensed clusters. And with all 3 genomes, you get even more condensed clusters. Of course, ideally, you would like to have those, but you mostly end up with those that detect more than one genome. The other point is, when you do the genotyping and you order the markers according to their genetic position. So up here, you have the lines. Down here, you have ordered the markers according to their position in the genome or according to the position at the genetic map. You clearly see very clear patterns. For example, here you have only 2 different patterns. We also call that haplotypes. And in the fact, all these markers in these regions, and there's way more than 50 markers in these regions, all of them create redundant information. So you don't need to generate this information 50 times. You can reduce that by only taking a few markers from these regions, and then use that. And if you do that in an optimized array, you see here that you lose these large haplotype blocks. You still get the information, but you get that information only with 2 or 3 markers. The other point is, of course, you want to know where the markers are located in the genome, so you want to select them. You want to locate the markers according to their position in the genetic map. Because it's known that recombination is accumulated towards the end of the chromosome, so you don't want to order or position the markers based on physical maps. You want to position the markers based on genetic maps. And of course, as I said, known genes. Some of the main genes in the wheat genome are various protein quality, reduced height, photoperiod sensitivity, vernalization genes, for which you want to have diagnostic markers also on the array to see what kind of phenotype you can expect when you make predictions on these traits. So version 1, 2.5 years ago, was about 13,000 markers that are of high quality that can be used. There's more markers on the A and B genome and fewer on the D genome, which has historical reasons. The D genome in wheat is the least polymorphic. The other genomes are more polymorphic. So almost all of those markers are perfectly callable. They are have a decent allele frequency, so more than 95% of the markers, for example, are polymorphic in some of the lines in European material. Interestingly also, although this was not intention, more than 50% of the markers are also useful in the tetraploid spaghetti or durum wheat material. Of course, also the 90K array with costs of 100 or over 100 euros for everything, including processing and data analysis, could be reduced by the smaller array to about 1/3 to 1/2 of the costs without losing too much information, or essentially very little information. The customers we have were very excited about those arrays, but always when they look at the data, their attitude was, oh, there's certainly a way to improve these things, and there is more possibilities to do these things. So last year we started with version 2 of the wheat array, and in order to do that, we used another array which has been developed in the UK. It's an optimized, it was also claimed to be an optimized array derived from a large set of molecular markers, namely from an 820K AFI matrix array. On that array, there are 35,000 SNPs markers. We have genotyped that for at that time for 768 lines representing the respective gene pools, and have that then subsequently analyzed further. Here are the numbers. The array has, as I said, 35,000 markers. Out of those markers, the top 2 lines in my display, the polymorphic high-resolution markers and the no minor home markers, are markers that give clear clusters. We usually claim or we think that these markers are the best ones. The other ones in the different other classes need usually some improvement. So out of the 35,000 markers, we have about 20,000 markers that are useful. As I also said, the main issue is that you need to target not individual markers, but you need to target haplotype blocks. So we have done the data analysis, and with the 35K array, we have also seen that if you look in certain specific regions, you have large haplotype blocks to some extent with many hundreds of markers that detect the same haplotype block when you look over all the varieties. Typically, there are some haplotype blocks with few markers, but there's also a considerable amount of markers that detect, create redundant data by detecting the same haplotype blocks. When you compare the 90K and the 35K array, the 90K array has about 11, or close to 12,000, detects 12,000 different haplotype or recombination blocks, and the 35K array about 10,000. There is a considerable overlap on the blocks, but there is also another 5,000 from the 35K that are not covered by the version 1 of the optimized wheat array. Also, there are some additional markers on the D genome that have been added to the new version. Again, what we also have added, I don't want to go in detail, but I just want to show you some more markers for known genes that are listed here. Some markers where we have data that they're linked to specific disease, quantitative disease resistances like fusarium or heading date or various other traits that are of relevance during the wheat breeding. So version 2, which is currently available, has now about 17,000 markers that had gone into the development of the array, and the analysis can now be done for 16,263 markers. The transferability from AFI to Illumina, to the Illumina platform, is a little bit limited because they have different requirements. So there is a certain failure rate based on the transfer from Affymetrix to Illumina. So that's version 2. That's what we have currently available. Of course, as I said, it's a continuous improvement process. So we're currently working on version 3 of the optimized array. Nowadays, in wheat, due to the very large genome size of 16 gigabases, what you do for polymorphism screening is you use not whole genome sequencing, but you use an available exome capture array, which is available from Nimblegen, and we have used that array to further identify interesting markers to look on the situation whether we can identify additional informative markers for the analysis. So the first experiments that we had done is we did sequence capture of 25 selected wheat lines, which we knew from the array-based genotyping that they are interesting. We are currently doing exome capture of another more than 200 lines where we have additional information. But the next slides I'll show you is based on these 25. So altogether in these 25 lines, we have identified more than 3 million SNPs. And then they went through several screening procedures, which I don't want to go into detail. 630,000 markers were then checked for suitability on the Affymetrix platform, and we went through other screening procedures. And at the end, we developed a 135K array. And again, looking only at the two major top classification, there are about 80,000 high-quality molecular markers that can be used. Again, we did the genotyping of the material in mapping populations, which is shown here. These are the mapping positions on an integrated genetic map for 41,000 molecular markers derived from 4 different populations. And again, the haplotype analysis on genotyping, a total of about 1,000 lines, and putting these things together. And again, in this, in this Venn diagram, you see that there's a certain set of about 6,000 additional haplotype blocks that we had not detected with the previous arrays. This number is, I have to make a remark, is overestimated because the data have not been fully cleaned, and so there are, it's probably an overestimation by at least 1/3 of actual haplotype blocks that we could add in version 3 to cover the entire wheat genome and to cover the entire haplotype blocks. So in summary, what we think is optimized genotyping arrays are ideal tools. They contain high quality markers. They have, if you, based on the large scale analysis, have a low ascertainment bias, which is confirmed since we have looked in various different types of germplasm. They are ideal for the breeding process, including genomic selection. Of course, as I tried to show you, the continuous addition of new markers increases the amount of information. We're still dealing with costs of 50 euros per sample when you analyze less than 100 samples, and less than 30 euros, including everything from DNA extraction to the final genotype table for routine analysis. It's a competitive price compared to other technologies, and it targets all the recombination blocks that you observe. I'm not going to talk about these things today, but we have developed the same things for maize and brassicas. And the success of these arrays can be shown that we're analyzing every year many 10,000 samples for breeding companies and increasing for academic research. So finally, the acknowledgements. There's a number of people from my company who have been involved in these things. We have a collaboration with CIMMYT on more exotic material, and you can analyze these things either through us or through LGC as our distribution partner. Thank you very much.